Who's Your Daddy and Mommy and Child by Catalina, Sophia, Natalie, and Leslie. Introduction. The objective of this lab is to offer students a comprehensive hands-on experience in the field of molecular genetics. By engaging in this experiment, students will deepen their understanding of genetic principles, particularly the inheritance patterns and molecular techniques used to analyze DNA. In this experiment, students will be provided with 6 DNA samples and tasked with determining the relationships among them. Specifically, they will identify which samples belong to the father, mother, and child. This exercise will not only reinforce key concepts such as DNA profiling, inheritance patterns, and genetic markers, but also introduce students to essential laboratory techniques like gel electrophoresis. By the end of the experiment, students will have gained valuable insights into how genetic testing is used in fields such as forensic science, medicine, and genealogy, preparing them for more advanced studies in genetics. Background and significance. Gel electrophoresis is a well-established and widely utilized technique in molecular genetics with significant applications in various fields, including biological relationship analysis and forensic investigations. This technique involves the separation of DNA fragments based on their size by applying an electric field to a gel matrix. DNA molecules, which are negatively charged, migrate towards the positive electrode, with smaller fragments moving more quickly through the gel than the large ones. This differential movement results in the formation of distinct patterns or bands, allowing scientists to visualize and compare DNA samples. One of the most critical applications of gel electrophoresis is in paternity testing, where it is used to analyze genetic markers known as short tandem repeats, or STRs. STRs are specific sequences of DNA that repeat multiple times in a row at various locations in the genome. Each person inherits half of their STRs from their mother and half from their father, making them highly effective for determining biological relationships. By comparing the STR patterns of a child with those of the mother and the alleged father, scientists can either confirm or exclude paternity with remarkable accuracy. Beyond paternity testing, gel electrophoresis plays a pivotal role in forensic science, where it is employed to identify individuals through DNA evidence collected at crime scenes. DNA profiling, which involves the analysis of unique genetic markers, has revolutionized investigations by providing reliable evidence that can link suspects to crimes or exonerate the wrongly accused. In the realm of genealogy, gel electrophoresis is used to trace ancestry and study population migrations over time. By examining DNA variations among different populations, researchers can map out historical migration patterns and uncover ancestral lineages. This has led to a deeper understanding of human history and the connections between various ethnic groups.
The hypothesis we will test in this experiment states that if more of the DNA samples are collected directly from the participants rather than a toothpick swab, then the data given by the gel electrophoresis will be more accurate in determining who the parents and the child are from the samples. This is because their observed bands in the gel will stand out with similarities and help us to decipher who is related. The purpose of our study is in aiming to boost student engagement by combining DNA analysis and gel electrophoresis into a hands-on experiment. Instead of just analyzing pre-generated images like in our own lab, students will be able to prepare DNA samples and run them through gel electrophoresis and interpret band patterns to identify family relationships. This interactive approach will enhance their understanding of gel electrophoresis and genetic analysis. In terms of our experiments, design, and methods, the revised molecular genetics lab aims to enhance the accuracy of DNA band analysis using gel electrophoresis to determine genetic relationships. In this revision, the distance migrated by DNA bands in centimeters is the independent variable, while DNA band size in base pairs is the dependent variable. Room temperature, procedural steps, and mixing techniques from the original lab will remain constant to ensure reliability. The primary modification involves sample collection, where participants will provide DNA by spitting into a cup. From this, 1,000 microliters, which is the equivalent of 1 milliliter of DNA, will be extracted using a micropipette, then centrifuged and integrated into the existing protocol. This change standardizes sample volumes and enhances band visibility, leading to more precise results. Procedures 1, 2, and 3 from the molecular genetics lab manual are combined to analyze DNA similarities and relationships effectively. This streamlined approach maintains consistency while improving clarity and accuracy in genetic analysis. In terms of lab safety in our new molecular genetics lab, some safety precautions that we will need to follow are wearing gloves to avoid exposure to harsh chemicals that may damage our skin barrier at all times. Remove and clean exposed jewelry and eliminate potential water contact with the electrophoresis unit. This includes any water bottles that we bring to the lab. And we need to ensure that the power button is in the off setting when handling the gel because of how the electrophoresis unit is sensitive at high voltages, and we need to make sure not to touch the electrodes on the electrophoresis units. We need to confirm that the amber filter is in place to protect our eyes from the UV light emitted, and we never leave the electrophoresis units unattended during operation. And done.
With the method of our experiment, it is important to recognize that the steps and procedures involved in gel electrophoresis are meticulous and can be quite time-consuming, as we found out. This process begins with the collection of saliva samples from 6 individuals, which are provided by the TA and labeled 1 through 6. To prepare, you must obtain 6 microfuge tubes and label them correspondingly 1 through 6. And once you do that, you're going to add 500 microliters of a 10% shellac solution to each microfuge tube. Shellac is crucial in this context as it functions to remove metal ions that could otherwise activate DNA degrading nucleuses, which protect the integrity of the DNA sample. Once each microfuge tube contains 500 microliters of shellac, carefully add 2 microliters of the corresponding DNA sample into each tube. Ensure the tubes are securely closed and proceed to incubate them at 95 degrees Celsius for 10 minutes. This heating step is essential as it lyses the cells and helps deactivate nucleuses, which prevents DNA degradation. During this process, it is critical to keep track of each tube using the appropriate label to avoid any mix-ups, because that could be bad. And after you heat the sample up, cautiously open the tube and release any built-up pressure, and note that the tubes may be hot, so please handle with care. Following the pressure release, securely close the tubes again. Next, balance the microfuge tubes with those of a lab partner and centrifuge them at a maximum speed for 10 minutes. This step separates the shellac and cell debris from the DNA, allowing for the collection of DNA containing supernatant. After going through the centrifuge, carefully transfer 8 microliters of the supernatant into a PCR tube containing 10 microliters of 2X Fusion Flash PCR master mix. Take special care in avoid aspirating any Shellac beads, as their presence can inhibit the PCR process. To each PCR tube, add 2 microliters of the forward and reverse primer from the microtube labeled P, and this will ensure the final reaction volume is 20 microliters. Then gently mix the contents by tapping the PCR tube with your index finger, and then spin down the reagents using a mini centrifuge at the bench. Confirm that the PCR tube cap is tightly closed before placing the tube back into the DNA thermocycler. Then you're going to select the Fusion PCR Program 980 and initiate the run. Upon completion of PCR amplification, vortex the PCR tube gently and spin down the contents again using the mini centrifuge. Ensure that all group members are prepared before proceeding to load the gel. Carefully remove the plastic comb from the gel wells and discard it. Immediately and meticulously load 10 microliters of each PCR sample into the corresponding wells, matching the well number with the tube number. Fill wells 7 through 9 with 10 microliters of distilled water as controls, and then once ready, obtain the marker from the TA and load 10 microliters of that into lanes M and 10. After all the samples have been loaded, connect the power cord and place the lid securely on the electrophoresis unit. Then you're going to select the 1 to 2% E-Gel X setting on the iBase and press the go button to initiate the run. The screen will indicate when the run begins. The electrophoresis process will typically take about 10 minutes. To monitor the migration of the samples, press the light button on the screen. However, ensure that you do not view the gel without the lid in place. As Sophie said, this can cause serious eye damage. The run will conclude automatically as indicated by the flashing screen and rapid beeping sound. At this point, consult your TA to determine if any additional runtime is necessary to achieve good and accurate, clear results. And with that, that is the method of our experiment. Thank you.
The materials and equipment needed for a genetics biolab is our gel electrophoresis, which looks something like this, which enables us to compare and contrast different DNAs. And we will also require our micropipette, which will be this one, which must go up to 1,000 microliters for our DNA samples. And we will need our DNA markers and our 500 milliliters of the 10% Shellac liquid. We will need our microcentrifuge tubes, which are like this, to do the DNA mixing and for the microcentrifuge, which looks something like this. And we will need our distilled water and our Fusion Flask PCR master mix, and we will need 10 milliliters of that. We will need our thermocycler, which looks like this, which we will place our microcentrifuge tubes in. And we will need our forward and reverse primer, and we will need an incubator, which looks something like this. And that will all be provided by the lab coordinator. And as for things that will be provided by us, it will provide 6 DNA samples, 3 of which will be related, and 3 of which will be 3 others. And we will need at least 1 milliliter of each DNA sample, which equals to 1,000 microliters, which is why we need the micropipettes that go up to 1,000 microliters. And that's all for our materials and equipment. Now, as for our genetics biolab predicted results, the results should be definite due to there being 3 related DNA samples and 3 other samples, and that is an unchanged variable, and so this is why our results should be definite. However, if there is a mix-up, or if the procedure is done incorrectly, that would be on the fault of the researcher and not due to our samples. And if the outcomes are different, then the procedure will have to be done again in order to receive the correct outcome. And the only reason why it would be on our behalf as providing materials would be if the other 3 DNA samples were miraculously related to the original 3, which we would be able to find out through this lab, which would be pretty cool. And yeah, and here is what we predicted our results would be. We crafted a estimate of what our EGL would look like, and this is it. And our column 1 would be the child, our second column would be the mother, and the 5th column would be the father.